Welcome back to this next video in which we are discussing HPVs and cervical cancer. Okay, so in this next video what we want to discuss is how do you actually develop squamous cell carcinoma from having an HPV infection of the squamous stratified epithelium of the ectocervix. But before we move on to that, let me just give you a brief summary of the HPV life cycle, because it's really important that we all completely understand that before we move on any further. So remember, the HPVs, they infect cells in the basal cell layer of the squamous stratified epithelium, and infected basal cells then divide and produce daughter cells that will move up into the midzone, which will also be infected with HPV genomes. And then in the midzone, these HPV infected cells will divide very, very rapidly and will amplify the HPV genome hugely, so they'll all end up with nuclei stuffed full of HPV genomes. And then as they move up into the superficial zone, they will start producing the capsid proteins for the HPV virion particles, and they will encapsulate the viral genomes in the capsid proteins to produce full virus particles. And then by the time you were talking about the most superficial layer of the superficial zone, these cells will be full of HPV virus particles, and then when they're shed off, those shed cells will be full of HPV virus particles, and then when they're exposed to mechanical forces, they'll break open and the HPV particles will then be released. So the important thing that I want to stress from what I've just said is that unless you get rid of the HPV infections in the basal cells, then you can't stop this from happening. Until you've got rid of the HPV infected basal cells, then they will just continue to produce more midzone cells which will be infected with the HPV genome, and therefore overall the epithelium will just be producing HPV virus particles all the time. We've also discussed at the end of the previous video about how immune responses to clear HPV infections from squamous stratified epithelia are extremely slow, mainly because HPV infection of a squamous stratified epithelium is not life-threatening. It's not going to acutely endanger your life at all. So because it only affects the epithelium and doesn't go into the bloodstream or any other tissue. So that's why the immune response is extremely slow, and we know that generally about 90% of HPV infections will be cleared within 2 years, and then the other 10% will persist for much, much longer. And we know this from just general knowledge. Warts can last for years and years and years, so HPV infections can last a long time. So let's now talk then about how HPV infection of the squamous stratified epithelium of the ectocervix can then lead to squamous cell carcinoma of the cervix. So SCC, I don't know if I've told you this before, but that's the abbreviation for squamous cell carcinoma, and we're talking specifically about squamous cell carcinoma, of course, of the cervix. So how does this all begin then? Well, of course, we know that it has to begin with an HPV infection, and this will be a mucosal HPV infection, of course, and it will be most likely transmitted to the girl by sexual activity. So it begins then with an HPV infection, and this HPV infection needs to be a persistent HPV infection. So it needs to go on and on and on to actually cause a problem. So this needs to be a persistent HPV infection. Now, of course, there are two different types of mucosal HPV that could cause a persistent HPV infection, and those are the low-risk HPVs and the high-risk HPVs. So let me now just discuss what happens in each case. So you can either get a persistent HPV infection of the ectocervix squamous stratified epithelium with a low-risk HPV, or with a high-risk HPV. Now, if you get a persistent infection with a low-risk HPV, this does not cause cancer. This is not a problem, so this is no problem at all. It's if you get a persistent HPV infection with a high-risk mucosal HPV that you actually move along the process to getting squamous cell carcinoma. And the reason is that the high-risk HPVs, if you have a persistent infection with them, they do something, or something tends to happen that doesn't happen with the low-risk HPVs, and this is that you tend to get integration of the genome into the host cell genome. So the key thing that happens in the case of high-risk HPVs, but not for low-risk HPVs that are causing persistent, prolonged infections, is that you get integration of the genome. And this is a really important new concept, and therefore I'm going to spend just a little bit of time to make sure that everyone completely understands what I mean when I say integration of the genome. So let me just draw a cell here. And let's say that this is a mid-zone cell of the squamous stratified epithelium. So here is its nucleus, and let's put in an HPV genome. And just to make this basic, we'll just have one HPV genome, but in reality there could be far more. So we know that normally that HPV genome is not integrated into the host cell genomes. It exists as a separate little circle of DNA called an episome. It's not integrated into a chromosome, unlike, for instance, with HIV, which does integrate its genome into the host cell genome. However, if you have a persistent HPV infection, then eventually, and it's believed to occur by chance, it's still controversial how exactly it occurs, but one of the major theories is that it just occurs by mistake. What can happen by mistake is that you can end up in one of the mid-zone cells getting an HPV genome implanted into the human chromosome. So what I mean is, if I draw a little human chromosome, so let's say that one in pink is a human chromosome, what can end up happening is that the HPV genome can end up implanted into the human chromosome, like so. Okay, this is what I mean when I say integration, and this causes a major change that then starts you on the process towards squamous cell carcinoma. So before I explain why it leads to the process towards squamous cell carcinoma, let's just discuss why we think this can happen in the case of high-risk HPVs, but not in the case of low-risk HPVs. So remember the difference between high-risk and low-risk HPVs. The high-risk HPVs, they have a more powerful E7 protein. So E7 is more powerful, and therefore the overdivision that is caused by the E7 in the mid-zone cells is worse in the high-risk HPVs compared to the low-risk HPVs. In addition, remember the high-risk HPVs, their E6 protein has the ability to completely turn off the p53 pathway. And remember, the p53 pathway is important for activating DNA repair when DNA damage occurs. Right, so having cells that are over-proliferating really fast, that puts you at risk of getting DNA damage. If you have cells dividing really, really fast, then because division is such a complicated process involving, lots of things happening with the DNA, there is big risk in division that you do get DNA damage occurring. So dividing very, very fast puts you at high risk of getting DNA damage, and having the p53 pathway turned off means that if you do get DNA damage, it isn't repaired, basically. And this is the reason we think that high-risk human papillomavirus infection carries this risk of integration, because we believe that this happens as a mistake. The viral genome gets damaged and cut into a linear piece of DNA, this chromosome gets damaged and cut into two pieces, and then maybe the viral genome slots in the middle. So we think that it probably happens by mistake, and it's much more likely that it will happen if you've got a high-risk HPV than if you've got a low-risk HPV, because of these factors here. The fact that we are over-dividing, making DNA damage more likely, and that we have the p53 pathway turned off, which means that the DNA damage won't be repaired. So, going back up to my picture up here then, if you have a high-risk human papillomavirus infection of the squamous stratified epithelium of the ectocervix for long enough, we think that in one of the mid-zone cells where you've got E6 and E7 expressed, eventually what will happen is that you'll get integration of one of the viral genomes into a host cell chromosome, and that cell in which this occurs, and I'll just circle one of them that we can imagine is going to be the one. So let's say it's this one here that occurs in. Okay, that cell in which this integration event occurs is then going to be the one that takes you along the way to squamous cell carcinoma. And this is the reason that low-risk HPV infection doesn't carry the same risk, because the integration event doesn't happen. Okay, you can have a persistent HPV infection for as long as you like, and you won't get the genome integrated into the host cell DNA in one of the mid-zone cells, because of the fact that the E7 is less powerful, so they're not dividing as fast, and they have a healthy p53 pathway, because the E6 protein doesn't do the same thing in the low-risk human papillomaviruses. So, overall, what we've said so far then is that if you have a persistent infection with a mucosal high-risk HPV infection of the squamous stratified epithelium of the ectocervix, then eventually what will happen in one of the mid-zone cells, and this will take a long time to happen, eventually what will happen is in one of these mid-zone cells that is dividing very rapidly and has inhibited p53 pathway, because of the viral life cycle happening inside it, you will get occurring in that cell the integration of one of the viral genomes into a host cell chromosome. And this causes a crucial change in this cell now. And the reason is that now that the viral genome is implanted into the host cell chromosome, I'm just trying to think of a nice colour to go to now, I think we'll go to purple. Now that this has occurred, you do not get the same process occurring as before. Remember, before, what happened is eventually you had that early to late switch. The early proteins stopped being expressed. The E6 and E7 proteins, their levels went down, and that was when the cell was able to stop dividing really fast and commence the differentiation process once again and go up into the superficial zone, where it would start producing the late proteins and packaging the viral genomes up into capsids. That doesn't occur in this cell that now has the now has the viral genome integrated into one of its chromosomes. So you don't get the early to late switch. So the early to late switch breaks. So this breaks, and that means that the cell just continues to express E6 and E7 continuously. So E6 and E7 remain high forever now. Okay, now that it's got this viral genome implanted into the host cell chromosome, this bit here will continue to code for E6 and E7 at a high level forever, and therefore you don't get the early to late switch, you don't get this cell differentiating, it just remains in the mid-zone and divides and divides and divides and divides. And this can now give rise to squamous cell carcinoma, and let me explain why. Because if we think about this population of cells, and I'll just go back up to my drawing here, so let's have this cell here that I've circled in red. Let's say this is the one where the integration event has occurred. It's now just going to divide and divide and divide and divide, so I'll put some more ones that it produces here. So here's another one, and all of its progeny, they'll all be identical to it, so they will all have the implanted viral genome as well. So that's supposed to represent the fact that it's divided and divided and divided, because of course it overproliferates due to the E7. So it produces loads of progeny that are all genetically identical to it. They all have the implanted viral genome in one of the chromosomes. So it has produced a whole population of cells that will just continue to overproliferate and which are also genetically unstable. They have no p53 pathway because of the E6 protein. Therefore, they cannot activate DNA repair mechanisms when DNA damage occurs. So they are genetically unstable. Now, if it's worked, and indeed it has worked, I managed to copy this diagram that we drew long ago for the neoplasia process. Remember, in the neoplasia process, we said that mutation 1 was something that made you overdivide, and mutation 2 was something that made you genetically unstable. So here is clone 2 of cells that are overproliferating and genetically unstable. We have now got, in the mid-zone of the squamous stratified epithelium of the ectocervix, a population of cells that are all overdividing and which are all genetically unstable. We are at this stage of this diagram, clone 2. In this population of cells now, the neoplasia process can continue. More mutations can accumulate, and you can go along the process to neoplasia. So I hope you understand what the persistent high-risk mucosal HPV infection has done. It's done the first two mutations effectively in this pathway. It's started the neoplasia process. That's not finished it. This is not cancer yet. These cells with the implanted human papillomavirus genome are not cancer. They are on their way to becoming cancer. They are on their way in the neoplastic transformation process. So, these cells are on their way in the neoplasia process, so they'll just continue the neoplastic process now. And what the persistent HPV infection did is it started that whole thing off, whereas with the low-risk HPVs, that didn't happen because you don't get the integration of the genome as you do with the high-risk HPVs. The integration of the genome is the really important thing, and it takes a long time. This is why you need a persistent HPV infection, because if it's cleared within 2 years, in those 2 years, it's probably the case that the integration won't have happened yet. It's a rare thing for this to have happened, so it takes a long time of producing loads of these mid-zone cells with human papillomavirus genomes in them, and which are over-dividing and genetically unstable with an inhibited p53 pathway, for you to actually eventually get one in which the HPV genome ends up integrated into the host cell chromosome. But once you've got that, then you're on your way to the neoplasia process. Okay, so that is then the story of how we believe squamous cell carcinoma can arise in the squamous stratified epithelium of the ectocervix following persistent high-risk HPV mucosal infection. What I now want to say is go back to the facts that we discussed earlier, which is we're now beginning to understand how high-risk mucosal HPV infection that's persistent on other mucosal squamous stratified epithelia can lead to cancers developing there. The exact same thing that I've just described here can easily, well, it's easy to imagine how it could occur on another squamous stratified epithelium besides the one of the cervix that we've been discussing so much. And indeed, we do think it does happen. So we do think that persistent high-risk HPV infection of other tissues such as the vagina, the preputial mucosa, the urethral lining, the anus lining, and also the oropharyngeal mucosa, we think that that can lead to cancer emerging in exactly the way that I've just described here, i.e. it starts the neoplasia process, it gives you the first two mutations of the neoplasia process. And indeed, we even think that cutaneous HPVs, you probably have some of those that are high risk as well, and which, when they infect the skin, and are persistent infections, they can start the process towards skin cancer as well. But that's an area that we're still researching and which may develop much more in the future. Okay, the next thing that I want to talk about is the grading of cervical lesions. So the SIN1, SIN2 and SIN3 grading. And then I'd like to talk about screening for cervical cancer, the Papa-Nicolau test. So let's just talk about grading then. So there are lots of different grading systems used for cervical dysplasia. So this is an important word. Dysplasia means strange cell division. It means cell division that is probably aberrant, that is probably caused by cells that are part way along the neoplasia process. So coming back to this picture over here, these cells in the later clones in the neoplasia process, they'll be dividing very strangely. They'll have aberrant division. And when you have a bunch of cells that are dividing aberrantly, this is called dysplasia. Now, in the cervical squamous stratified epithelia, you are going to get cervical dysplasia, and we grade this cervical dysplasia into 3 different grades. And these are known as SIN1, SIN2 and SIN3. So I just want to explain this grading system. So firstly, what does SIN stand for? It stands for cervical, that's the C, and then intraepithelial, so within the epithelium, and then the N is for neoplasia, so it refers to the fact that the neoplasia process is occurring. So how do you actually decide whether the squamous stratified epithelium of someone's cervix is SIN1, SIN2 or SIN3? Well, you have to take a sample, and we'll talk about the screening mechanism later on, and you have to look at the cells, and you can see when cells are dividing aberrantly, because the nuclei of the cells will be different sizes and the size of the cells will be different as well. So in a perfectly healthy, and I'll go back up to this picture here, in a perfectly healthy squamous stratified epithelium of the ectocervix, all of the cells at the different layers, so for instance all of the cells in the superficial zone, all of the cells in the mid zone in particular is the zone that we're particularly interested in, and all of the cells in the basal cell layer, they'll all be the same size and they'll all have the same sized nuclei. Now, when you've got, for instance, the developing of a squamous cell carcinoma in the squamous stratified epithelium of the cervix, so let's say for instance that we have had a persistent high-risk HPV infection and we have now had it integrate its genome, so we do now have this red population of cells. These cells will be dividing aberrantly now, and you'll have cells of different sizes and with different sized nuclei, and that's how you know that dysplasia is occurring. So I might just draw a vivid example of this. So it might look like this. Here is one with a very big cell and a very big nucleus. Here's another very small cell, very small nucleus. Here's another big cell and it might have a tiny little nucleus. So you've got very odd arrays of cells. They don't look similar to one another. They're different sizes, different shapes, and they have different sized nuclei. This is the way that you see that the mid-zone cells are dysplastic, that dysplasia is occurring there. And you grade it according to how much dysplasia you think is there. So the grade cervical intraepithelial neoplasia 1, this is the grade of dysplasia that you would get if you just have a persistent high risk, well, not even a high risk, a persistent HPV infection. So if you've got a persistent HPV infection in the squamous stratified epithelium of the ectocervix, then the mid-zone cells will be dysplastic. They will be dividing aberrantly because the human papillomavirus is using its E7, and if it's a high-risk one, it's E6 as well, to make the cell divide really rapidly, and therefore you will get some degree of dysplasia just from having a persistent HPV infection. This is before you've ever had integration of the genome. Just by having the persistent HPV infection, or even a non-persistent HPV infection. So I should put that there. So this is really just having an HPV infection present within the cervical squamous stratified epithelium. It will cause the mid-zone layer of cells to be dysplasic. They will have different size nuclei and different shaped cells, but it won't be as extreme as these higher grades. So it won't be quite this bad that I've drawn here. I've drawn a really extreme example here, but you will see mild dysplasia there. Okay, coming up to grade 2, cervical intraepithelial neoplasia grade 2, that's the sort of grade that you will see once you've got integration of the genome. So I'll put here integration. So if you've got a low risk human papillomavirus infection that is persistent, you will never get that developing into SIN2. It will always just stay a SIN1 lesion rather than a SIN2 lesion. If you've got a high-risk human papillomavirus persistent infection, then eventually what might happen is integration of the genome, and then you'll get worse dysplasia occurring in that mid-zone, and that will now be SIN2, so you'll see more dysplasia, you'll see more radically different sized cells and more radically different sized nuclei in SIN2. And then SIN3 is where it's getting further along the neoplasia process, so this is further along the neoplasia process. So what will have happened is you will have more advanced clones of cells. So remember, once you've got integration, you've then got permanent expression of E6 and E7. So you're effectively clone 2 in the neoplasia process. Once you've got more of the mutations and you're a higher clone, you're worse now, you're closer to being cancer, then the dysplasia will be so severe that you'd grade that as SIN3. Above SIN3, there is then carcinoma in situ, which we've discussed previously. This is the same as cancer in situ, but we can now call it specifically carcinoma because we're talking about a cancer of an epithelial tissue, which is a carcinoma. So CIS is above CIN3. This is carcinoma in situ. This is basically, you've got a clone of cells that is on the point of being cancerous, but they have not yet gone through the final mutation that will give them the ability to invade the surrounding tissue, so it is in situ at this point. And then, of course, after that, after you've acquired the final mutation that makes you invasive, it will then be squamous cell carcinoma. And we, of course, then have the 4 different stages of squamous cell carcinoma. Stage 1, where it's still in the cervix. Stage 2 and 3, where it's invading surrounding tissues such as the vagina, the uterus, the bladder, and the rectum. And then stage 4, where it's acquired another mutation that's further along in the microevolutionary process, and it's now metastatic. Okay, so this is the grading of neoplastic tissue in the cervix. It's the grading of how far along are the mid-zone cells of your squamous stratified epithelium of the ectocervix in the neoplasia process, and it gets higher. So CIN1 is just having an HPV infection, CIN2 is when you've had integration of the genome, CIN3 is further along the neoplastic process but not quite at the point where we're carcinoma in situ yet. Then we go up to carcinoma in situ, and then we go up to squamous cell carcinoma. So that is the grading. Finally, there's another grading system that I just want to introduce you to because people use it regularly. So CIN1 lesions are also called low-grade squamous intraepithelial lesions, LSIL, and you do see this used, so that's why I'm just bringing it up here. So this stands for low-grade squamous, and then the I is for intraepithelial, and then the L for lesion. So low-grade squamous intraepithelial lesion. So this is another grading system for cervical dysplasia. And then CIN2 and CIN3 are grouped together in this other classification, and they're just called high-grade squamous intraepithelial lesions. So HSIL rather than LSIL. So CIN2 and CIN3 are combined together and they're called high-grade squamous intraepithelial lesion, and they are at risk of developing into squamous cell carcinoma. Okay, so let's now talk about screening techniques, and this of course will make it more obvious what we're talking about when we talk about these gradings of cervical neoplasia. So screening now. So we screen women for cervical neoplasia, we screen them for developing cancerous lesions. We can see under the microscope how far along you are in the cervical process. We can see the neoplastic process. We can see developing lesions that will eventually become squamous cell carcinoma. And at that stage we can do something about them, so it's a very good idea to screen for the developing lesions. So this test is named after a Greek doctor by the name of Papanikolaou. So let me just struggle with the spelling of this and then we'll go on. So Papanikolaou, spelt like so. So this is called the Papanikolaou test, more often just abbreviated down to the PAP test. It's also called the cervical smear test, and this is a very simple idea. All you do is you go into the vagina, you go up to the cervix, and you take a sample of cells from the cervical squamous stratified epithelium. So you scrape off a few sample cells, you take those cells back to a laboratory, you look at them underneath a microscope, and then you are looking for dysplasia. You are looking for loads of different sized cells with different sized nuclei, and then you can grade the dysplasia as either CIN1, which is the sort of dysplasia you'd get just from having a persistent HPV infection present in the squamous stratified epithelium, or CIN2, which is once you've got integration of the genome and permanent expression of E6 and E7, and then 3, which is more mutations on top of that, which will lead to more of the properties of cancer, so it's a worse neoplastic lesion. Carcinoma in situ, where you've got all the properties of cancer apart from that you're not invasive, and then of course finally squamous cell carcinoma. Now, if you find that someone has a lesion that you would grade as CIN1, or a low-grade squamous intraepithelial lesion, then you do not do anything about this. This just means that they have a persistent HPV infection, and it may well be a low-risk HPV infection, and therefore it does not need to have anything done about it. If you find that someone has a high-grade squamous intraepithelial lesion, or even worse, carcinoma in situ, then something does need to be done about it. The lesion needs to be destroyed, basically. The cells that are this dysplastic need to be destroyed, and there are lots of different ways that this can be done. They can either just be surgically removed, or they can be destroyed by freezing them or by lasering them, destroying them with lasers. So there are fancy words for these techniques. So destroying the cells by freezing them is called cryotherapy. So you go in and freeze the cells to death, and of course that annihilates the lesion, and once those cells are dead, it might cause a horrible scar, but they're dead. It's not going to develop into cancer anymore. And the other technique, which is destroying the lesion by lasers, is called laser vaporization. So in that case, you laser the cells, you destroy them with laser energy, and again that destroys the lesion, and therefore if you've killed all the cells, then you stop the neoplasia process. If you've completely annihilated the clone that is on its way through the neoplasia process, then it goes back to square one, and you have to start the neoplasia process all over again. So you've stopped the process from occurring. Okay, so that is the purpose of screening for cervical lesions, so that we can see these precancerous lesions, these early neoplastic lesions that are not yet cancer. So I really want to stress this point. We can see the early lesions, the early neoplastic lesions, and because the cervix is so accessible, we can screen this, we can test it and make sure that these early lesions are not there, and if they are there, we can destroy them to stop them progressing any further towards being squamous cell carcinoma. Okay, finally I want to say a few little words about the treatment of squamous cell carcinoma if you do develop squamous cell carcinoma. Now I'm not going to say much about this. So if you've got squamous cell carcinoma, or indeed carcinoma in situ, and the squamous cell carcinoma is stage 1, which remember means that it is still just in the cervix, the tissue where it arose, then of course the treatment will be to have it surgically removed. If you remove all of the cells, then that destroys the disease. It can't progress any further. So you've cured the disease. So that's what's nice about stage 1 cancer, that if you surgically remove the tumour, then the disease is cured. For stage 2, stage 3, and then poor old stage 4 squamous cell carcinoma, then you cannot as easily surgically treat the disease. Instead, you have to use radiotherapy and chemotherapy as well. Radiotherapy, you use radiation to destroy the cells of the tumour. In chemotherapy, you use drugs that stop cells dividing, and I'll give you 2 examples of these. So I'll just put this here. So 2 examples of classical chemotherapeutic agents. And when we say classical chemotherapy, these are drugs that just stop cells dividing. They're a very, very blunt technique. So we're not talking about newer anti-cancer chemotherapy agents that specifically target certain signaling pathways to destroy the reason that the cancer is over-dividing. These are the classical chemotherapy agents, which are really very blunt. There's nothing clever about them. So it really is the case that, you take on the problem, you say, cancer is a disease where cells are over-dividing, therefore we will give a drug that stops all cells dividing. It's a very, very blunt approach to treating cancer, and that's how the classical chemotherapeutic agents work. The more modern ones that we're still developing, they are cleverer, and they say, okay, why are the cells over-dividing, and let's specifically target the reason that the cancer cells are over-dividing, rather than just throwing an agent that stops all cells in the body from dividing, which is what classical chemotherapy is. However, classical chemotherapy is still extremely useful, and it does save lives. It does obviously have severe side effects though. So 2 examples of classical chemotherapy agents that are used in squamous cell carcinoma are cisplatin and etoposide, and they both have different mechanisms. We're not going to go through the exact mechanism of these drugs. All that you need to know is the basics, that these stop all cells that they go into dividing. So you give these drugs, they will stop all cells in the body from dividing. That's fantastic because it stops the cancer cells from dividing, but they will have severe side effects. Okay, so classical chemotherapy and radiotherapy will be the treatments for stage 2, 3, and 4 squamous cell carcinomas. And if you're just going to learn 2 classical chemotherapeutic agents, these are 2 really important ones to know the names of: cisplatin and etoposide. So, we'll have a break here. In the next video, we will talk about the great success, human papillomavirus vaccinations, and that will be the final video.